good evening to our very special guest speaker, Scott Smith, who's dialing in all the way from the US today to give us a talk. So we're very, very grateful for you to take time out of your evening to do that for us. I'm just going to read a little bio and then hand over to Dr. Smith for the talk. And we'll have some time for Q&A at the end. So please do think of some questions to ask. Scott Smith leads the Nutritional Biochemistry Laboratory at NASA Johnson Space Center. This group is charged with keeping crews healthy with respect to nutrition, including using nutrition to optimize astronaut health and safety. This work includes ground-based and spaceflight research to understand how nutrition can mitigate the risks of spaceflight. Dr. Smith has ongoing research projects on the International Space Station. His past projects have been flown on the space station, Space Shuttle, and the Russian space station, Mir. Smith has also led several ground-based research projects to better understand astronaut health in space, including studies of vitamin D in crews in Antarctica, studies of crews living on the bottom of the ocean, and studies of test subjects spending weeks to months in bed. Dr. Smith is a member of the American Society for Nutrition, the American Physiological Society, and the International Academy of Astronautics. He holds a BS in biology and a PhD in nutrition, both from the Pennsylvania State University. So thank you again for coming along today, and over to you.
Thanks. Well, again, I appreciate the invitation, and I appreciate the opportunity to talk to you a little bit about what we're doing here. So again, my name is Scott Smith, and I lead the Nutritional Biochemistry Lab here at the Johnson Space Center in Houston. What I hope to do is to give you a bit of an overview of who we are and what we do, some of the work that we've done recently, and some of the work that we have coming up. Everything at NASA has to have a logo, and that's ours in the middle there. NASA logos are always overwrought with detail. Ours is no different, but you've got the human being here. You have food represent the interaction there. You've got an artist licensed vitamin D molecule on the side. The one carbon unit here points to Houston. And then you've got the space programs. You've got the space station up here, the moon where we're going next, and then Mars where we hope to get in the near future. So we try to cram all elements of what we do in here. I would like to start with this picture. This is back from 1992 now, 31 years ago. This is when we were celebrating the 500th anniversary of Columbus's trip from Europe, and he found America. And I use this to make the point that when we talk about nutrition and exploration, if we go back to the history books, there are many examples of how nutrition either made or broke exploration missions. The age of sail is no different. Scurvy is always a common thing that people would think of. And indeed, scurvy was a big deal, to say the least. Scurvy, vitamin C deficiency, as we know now, killed more sailors than all other causes of death combined. And as we look to explore beyond Earth's orbit, we need to make sure that we understand the nutritional requirements, the food system, the stability of the foods, how the body changes, and all those things need to be right, or it's going to be a bad trip, as we say. And that is sort of the backdrop under which we work. Our laboratory does two types of work. We do what we call operational work, or clinical work, if you will, and we do research. And I'll try to give you a bit of a feel for both of those. On the clinical side, we do a clinical nutritional assessment profile where before and after flight, we work with all the space station crew members. And before flight and after flight, we collect blood, we collect urine, and we measure a broad swath biochemistry, things like vitamins and minerals and proteins and bone markers and kidney stone risk, I mean, you name it. And we do that so that when we send crews up, they're as healthy as can be. We do that when they get back so that we can assess if there are any decrements, that we can get them back to full health as quickly as possible. And then during flight, we track body mass. And that picture in the middle there, one of the astronauts on a device called the SLAM-D. Everything has to have an acronym here as well. This is the Space Linear Acceleration Mass Measuring Device. And you can see Karen Nyberg there perched on it. It locks into place about a foot away from the wall. You hold on really tight, you push a button, and it pulls you towards the wall. It measures your acceleration, and using the physics of force equals mass times acceleration, we can calculate their body mass. So in other words, this is a scale. And when you think about it, when you go to the doctor's office, the first thing they do is put you on a scale, and that's because your overall body mass and changes in body mass give you obviously a very good index of whether or not things, again, in a gross sense, are going well or not. So we do track body mass during flight. The other thing we do is we track dietary intake, and we built an iPad app a few years ago that was used on station for about 6 years. This is known as the ISS Food Intake Tracker, or ISS Fit. You can see it floating there in the cupola on the left. On the right, you can see the iPad in Peggy's hands, Peggy Whitson, when she was up on one of her flights. And that is the login screen. So you pick your picture and you click on login. And what would then happen is you would come up to the screen on the left here. And when you start the day, the screen would be blank. But when you go to eat breakfast or when you've eaten something, you can use one of these buttons across the top here to record what you ate. So the button on the right is a quick hit for water. This is a search tool. There's a camera, so you can take a picture of the food. You can scan the barcode. You can leave a voice message with a microphone, or if you click on the folder here, you will go to the page on the right. And what you can see here is a long alphabetized list of all the foods that are available on Space Station. Here we've got meat and fish, and vegetables and sides highlighted. And what would happen is you would then click on the buttons here for what you ate, and then the add to consumption button on the top right would become highlighted. You would click on it. It would take you back to that page on the left, and you can see there foods that were entered that day. For each food item, it shows you the calorie content, sodium content, and the fluid content. And if you swipe, it will show you protein, carbohydrate, and fat. And then at the bottom, it gives you a running total of your calories, your sodium, and your fluid. For calories and sodium, those are two nutrients that we worry about the crews getting enough. And therefore, those bars started red. And when they've met the requirement, the bar will turn green. Sodium is the opposite. We worry about crews getting too much sodium. So the bar starts off at green, turns to yellow, and then eventually will turn red when you've met or exceeded your recommended intake for the day. This is an incredibly valuable tool. The crews really liked it. And I think a couple things. One is the fact that it was autonomous, the fact that they could enter the data and it would tell them literally how they were doing, the fact that it was real time so that at lunch they could see if they needed to eat more at dinner were some of the elements that made this extremely valuable. We asked the crews to record everything they eat for the entire time that they are up there. So for a cruise on a 6-month mission, we expect to get roughly 180 days of data. We realize doing it every single day is probably asking a bit much, but I can tell you in the time we had this app on board, we had data more than 90% of the days that we expected. So it worked really, really well. Not only does this help with each astronaut, but it also allows us to go back in after the fact and look at dietary intake patterns in relationship to changes in health. And again, I'll show you some of those data today. One of the other things that we do operationally is we help with new mission development. So as you may have heard right now, we're working on what we call the Artemis missions, which will take the first woman and the first person of color to the moon. Artemis is the overall program, but there's actually 3 elements to Artemis. That is the Orion capsule that will take the crew from Earth out to lunar orbit, the Gateway program, which is building a small lunar orbiting space station, and then there's the Human Life Sciences program, which will develop a lander that the crew will fly up in Orion, dock to Gateway, go into Gateway. 2 of the crew will get in the lander, go down to the moon, and then we'll come back up, at least in the early days, after about 10 days on the moon, and then eventually we'll come home on that capsule. You may be wondering what a nutritionist has to do with building rockets, and the reality is not much, but those rockets are required to take care of our crew. And one of the challenges of building rockets is that they are always over mass and they are always over volume. That is, the stuff that we want to fly weighs too much and it takes up too much space. And for some reason, when mass and volume are limited, it doesn't take long for somebody to look at the food system and say, well, food and water take up a lot of mass and a lot of volume. If we could just send less food and less water, we could make this easier. And one of my jobs is to fight with the engineers to make sure that we are sending the right amount of food that we feel that the crews need to make sure that they can sustain themselves, that they can function at least 100%, because we expect them to do extremely challenging jobs in extremely challenging environments on extremely challenging timelines. So, well, again, we're not helping to bend metal. We do weigh in pretty heavily when it comes to what will be done on these missions. So on the research side, we do 2 types of research. We do what we call ground-based research, and we do flight research. The ground-based research we do can be exotic, if you will. And the ground-based research, we're always looking for what we call analogs, ways that we can simulate aspects of spaceflight to allow us to test things on the ground before we do them in flight. So from right to left, when we wanted to look at how much vitamin D you needed to supplement astronauts with when they don't see the sun for 6 months. We went to where the sun doesn't come up for 6 months, which is we tested crews in Antarctica. We didn't test the penguins, but they're cuter than the people we did test. As you heard, we've done studies at the bottom of the ocean. We've got a habitat off the coast of Florida that crews spent 10 days to 2 weeks in, and we looked at things like oxidative stress and changes in vitamin metabolism, changes in iron metabolism, changes in red blood cell metabolism to see how those were affected by a hyperoxic, hyperbaric environment. When we want to look at things like changes in bone and muscle and eyes, believe it or not, we can put people to bed for weeks to months. And the inactivity associated with that, if we tilt you head down at a 6-degree angle, there's a fluid shift associated with that that can increase intracranial pressure and can impinge upon the eyes. And we're in the midst of a couple of studies right now where we're testing just that. And I'm going to talk a little bit more about the ocular issues, and so I'll come back to that. We've also done chamber studies where we will put people in an enclosed habitat for extended periods of time. The chamber you see there is what we call HERA, which is the Human Exploration Research Analog. And we did studies in there up to 45 days. And one of the studies we were involved in was testing if we fed people better, would they do better? We recently published those results, I think it was about 6 months ago now, and we continue doing studies of that nature. And I'll talk a little bit more about that as well. So we've done some, again, interesting and somewhat exotic ground research, but nothing beats flight research. We've had some tremendous opportunities over the years, and our research, in most cases, boils down to tracking dietary intake and collecting blood and urine samples. And you can see the pictures across the top there. There's Luca Parmitano on the left drawing Karen Nyberg's blood. Second picture is Nicole Stott putting those blood samples into the centrifuge to spin them down. The third image is a urine collection device that we use to collect urine. It typically is supposed to be stored in a Ziploc to prevent urine from leaking around the cabin, but one of the crew members took it out and thought it'd be funny to take a picture, so I have to use it. And in short, I'll tell you that there is a toilet on board space station that works a little bit like those on Earth, which is you go to the bathroom and it goes away. When you want to collect samples to bring home, it becomes a lot more challenging. So we use these devices, and the crew voids in the bag, and we collect a syringe of urine, and that syringe comes home, and that bag goes into the trash. It is jettisoned along with the rest of the trash. Blood and urine samples are stored in the freezer. You can see Chris Cassidy on the right there working with what we call the Melfi, which is the -80 laboratory freezer on ISS. And the samples will be stored in that freezer until they're brought home, and then we can follow them and go off and analyze them. Starting to look at some of the data on the left is a dietary intake graph. And this is looking at servings of fruits and vegetables per day. Each blue dot is a male astronaut, each red dot is a female astronaut, and the white dot is we looked at the foods that we fly and came up with the best menu we could come up with, and that is where that white dot falls. So what you can see is on average, crews get about 2.5 servings of fruits and vegetables per day, which is pretty far below the 6 servings of fruits and vegetables we wish they would get. But what you can see is that white dot is only about 3.5 servings a day. So it's not that they're not eating their fruits and vegetables, it's that we're not flying enough. And we've been working with our colleagues in the foods lab to improve that. We've also been doing a study to see if we flew more fruits and vegetables along with other healthy foods to see how the crew would fare. The graph on the right is biochemical data, but what we're looking at here is the comparison of fruit and vegetable intake on the x-axis with antioxidant capacity measured at landing. And what you can see is there's a positive correlation there. That is, crews that ate more fruits and vegetables had better antioxidant capacity. And in astronauts in a high radiation environment, oxidative stress is one of our bigger concerns. And as I often say, data on the right are the biochemical evidence that your mother was right and that you need to eat your fruits and vegetables. Putting all this together, when we look at nutrition and bone health, but really nutrition and health in general, we've shown that omega-3 fatty acids are beneficial in protecting against bone loss. We've shown that diets higher in fruits and vegetables are better for your bones. We've shown that high iron stores, which is one of the things that happens during space flight, high iron stores are detrimental to bone, and that high sodium intakes are also detrimental to bone. So if we were to look at an optimal diet, you would have more omega-3s, more fruits and vegetables, less iron, and less sodium. Testing that was the study that I talked about in the HERA chamber we did recently, and there was a flight arm of that study that we're doing on orbit right now. We have 3 of the last participating crew members on board space station. Picture in the top right there is Victor Glover. He was one of the earlier crew members. And what we did for that study is we provided participating crew members with extra containers of fruits and vegetables and fish to help see, again, if we fed them better, if they would do better. And that was one of the studies we've collaborated on with our colleagues in the food lab. And just to show you that everybody has their own logo, their logo in the middle there. One of the more exciting things we've got going on right now is another chamber study. And this is what we call CHAPEA. It's the Crew Health and Performance Exploration Analog. Again, everything has to have a logo, so you can see that there. And this is Mars habitat study that we are doing here in Houston. We 3D printed a habitat out of cement. You can see two of the rooms on the top left and the bottom left, living areas in the top left, and then two of the crew's bedrooms are in the bottom image. It's a 1,700 square foot habitat. It houses 4 crew members and has living area, it has work area, has an exercise area, and next door to it, there is a 1,200 square foot sandbox of red tinged sand that you can see in the bottom right. And the crew will be required to do a number of EVAs, extravehicular activity, where they will need to put on a simulated spacesuit and go out into the sand and either dig up rocks or put together different structures. There's a treadmill that you can't see, but it's out of the picture. We'll have to walk on the treadmill to simulate going distances from the habitat. This is by design a 378-day mission, and we're hoping to do 3 of these missions consecutively, not consecutively, in Syria. We started mission 1 2 weeks ago today. So today is day 14. That is the first crew. That is their logo there in the middle. And they only have 364 more days to go. Again, it's a Mars habitat, so communication is delayed. So they can't call anybody on Earth. They can email, but the computer systems are set up such that when they send an email, it takes 22 minutes for us to read it, and then we can type an answer and send it back, and it takes 22 minutes to get back to the crew. So the crew is fully autonomous. They've got all their supplies in that habitat. All the food that they're going to eat is in that habitat, and they need to get through this to make it through that 378 days. So again, we've been working towards this for about 5 or 6 years now, and the fact that we put them in that chamber just 2 weeks ago is incredibly exciting. As I said, there will be 2 additional one-year missions, and if anybody's interested, I'm guessing in the next 4 or 5 months, we'll start recruiting for the mission that will start probably next summer or next fall using our calendars. Late 2024 will be the next start. To dig into one of the areas of research that we've been working on, about 12 to 14 years ago, we realized that some of the astronauts come back from space station with changes in their eyes. There's some nuance to it, but one of the elements is that there are changes in the retina in the back of the eye. Specifically, one of the bigger concerns is what we call optic disc edema. So the optic disc is the point where the optic nerve enters the back of the eye, and that area swells and you get edema, which is obviously not a good thing. Everything has to have an acronym, so we dubbed this SANS, which I don't really like, but they didn't ask me, but they're calling it Spaceflight Associated Neuroocular Syndrome. And one of the key elements of this, in my mind, is that only about 20% of the astronauts develop SANS. So there's a lot of people think that you go into space flight, you're weightless, the fluid goes into your head, the pressure in your head goes up, and that pushes on your eye and causes this problem. If that were true, if it was that simple, everybody would develop this. And what we've struggled with all along is that not everybody gets it, and therefore there must be some predisposing factor which leads some people to get this, but not everybody. When this came about, we had biochemistry data on a lot of astronauts, and we started looking at our data to see if there were any differences between affected astronauts and unaffected astronauts. One of the things we were looking at was what's called one-carbon metabolism, which is a biochemical pathway that occurs in every pathway, every cell in your body. It's called one-carbon metabolism because it's how the body moves single carbon units around. So it's how you methylate DNA, it's how you interconvert amino acid. And I often say that this is one of those things when you saw it in school, the first question was, is this on the test? And it turns out in real life, this thing is on the test. HCY in the middle there is homocysteine. And homocysteine is an amino acid metabolite. If you are folate deficient, if you're B12 deficient, if you're B6 deficient, your homocysteine levels will go up a lot. When we started looking at our homocysteine data, we found the graph on the right, which shows that astronauts that developed ocular problems, that is those in the yellow line, had significantly higher concentrations of homocysteine in the blood compared to astronauts that did not develop ocular problems. And most importantly, these first 3 data points were before flight. So we went from this initial finding, we hypothesized, given there were differences before flight, given we know that genetics affect one-carbon metabolism, affect homocysteine, that this may be genetically predisposed. We did 2 studies. One was a spaceflight study, one was a bed rest study, looking at different single nucleotide polymorphisms. And very briefly, to look at these data, the graph on the left shows you one of these SNPs, MTRR A66G, and this is an methionine synthase reductase enzyme. And what we found, again, here, every dot is an astronaut. And what we found is that every astronaut that had the minor form of this one SNP developed ocular findings. And that is to say, nobody that didn't have that SNP developed ocular issues. These are data we first broke out in 2014, and it was around this point that some people started to think that there might be something here. Some people besides us, I should say. We then did a bed rest study, and they put 11 people to bed. They used a strict head down tilt protocol, and some of the subjects developed optic disc edema. On the y-axis here, this is the change in total retinal thickness, which is a quantitative measure of optic disc edema. And what we found is when we were looking at these two SNPs, that individuals that had more of the risk alleles had significantly greater optic disc edema compared to those that did not. So from this, we concluded that genetics and B vitamin status predisposed some individuals to develop SANS. One of the criticisms we got along the way was that, well, how can your genetics affect your eyes? Which is a bit ridiculous to say, but that's what we were told. We sat down and went off and looked at the literature and came up with a hypothesis that we've diagrammed here. What we think is going on is that your genetics affect your homocysteine, it affects your folate status, and in combination with some or many factors that we know occur during spaceflight, we maintain that the endothelial function is altered in those individuals. Specifically, that with low folate, your endothelial nitric oxide synthase is uncoupled so that you don't make nitric oxide. You end up with endothelial dysfunction or blood vessels that don't relax as well. You end up with increased oxidative stress, and that this can lead to edema. And that edema in and around the optic nerve can block the arachnoid villi, can block cerebrospinal fluid drainage out of the head, which can lead to an increase in pressure, which can push on the optic nerve and causing these ophthalmic findings. Another potential explanation is that we know increased peroxynitrate will lead to an increase in matrix metalloproteinase activation. An increase in matrix metalloproteinase activation can cause an increase in collagen turnover and elastic breakdown. Those two things will make structural components in the body more stiff and less elastic. One of those components is the sclera, or the back of the eye. And what, again, we hypothesize is that in at-risk individuals, their endothelial biochemistry is different to the point where it affects the structure of the sclera, making it more susceptible to fluid shifts, making them more susceptible to these ocular problems. Now, again, what I've laid out here are two of our hypotheses of how this could be happening. I don't know this is right. We don't have data to back this up, but we know each of the arrows in there is supported by literature. So we know that scientifically all these things are plausible. Whether or not this is the end-to-end explanation of what causes SANS, nobody knows yet. A significant and not the most significant question remaining is whether or not we could use B vitamin supplements to either mitigate or prevent individuals who are at risk from developing SANS. We've got an experiment that we're about to start called B Complex. Again, everything has to have a logo. That's the one we built for this. And this is an experiment where we will give astronauts vitamins, specific combination of B vitamins, and we will look at that in relation to their genetics and hope to see that we prevent some individuals from developing SANS who we think might have done that. We have started pre-flight data collection. Our first astronaut is in this image here. The woman right there, third from the left, is Jasmine McBelly, and she will launch to space station on the SpaceX vehicle. She's part of what they call Crew 7, and they will go to the space station and become part of the Expedition 70 crew. So again, everything has to have their own logo. And she did her first pre-flight data collection back in March. She's been taking those supplements ever since. She'll continue to take them throughout flight and then for another 30 days after flight. And we're looking at ocular coherence tomography to look at her eyes. We're looking at her biochemistry. We're looking at her blood vessel function. And we're looking at advanced glycation end products, which are one of the markers of alterations in matrix metalloproteinase function. So again, this is an incredibly exciting time, and we are literally counting down the days for her launch to space station. Shifting gears briefly, I'm going to go to a topic that might seem a little strange, but let's talk about venous thromboembolism, or VTE, or blood clots, as I like to call them. When we talk about blood clot risk, You have to go back to Virchow's triad. And if you're not familiar with this yet, I'm sure you will be soon. Virchow was a 19th century physician who posited that there are three elements of blood clotting, and that your risk of a blood clot forming is related to these three things. That is hypercoagulability, alterations in hemodynamics, and alterations in endothelial injury or endothelial dysfunction. If you have all three of those things, then you're at the highest risk of blood clot formation. So why are we talking about this? Because a few years ago, we had an astronaut develop a blood clot during spaceflight. This was a letter sent to the New England Journal of Medicine, and you can read along where it says, 2 months into a space station mission, a left internal jugular venous thrombosis was suspected. It was found during ultrasound, and this was a little bit like the vision problem, that we didn't know this was a problem until we realized we had an issue. So as you can imagine, as the physicians taking care of the crew were trying to figure that out, they didn't call us, probably for good reason. But nonetheless, what we did after the fact is we started looking back at our data to see if there was anything of interest. And one of the things we started to look at was albumin. I'm sure you're very familiar with albumin. I would say albumin is the vanilla of proteins. That is, everybody knows it's there. You probably know it's half the protein in your circulation, and that it's kind of important. But albumin actually, I think, is a lot more important than most people realize. One of the things we know is that if your blood albumin concentrations or your serum albumin concentrations are low, then that will increase your blood viscosity. Because one of the things that happens is it changes the red blood cell fatty acid composition and will increase the viscosity of the blood. Albumin is also responsible for maintenance of the glycocalyx. The glycocalyx is the fine hair-like structure on top of the endothelium that lines every single vessel in your body, and that it's very fragile. And if your albumin concentrations are low, you will get glycocalyx degradation, and that equates to endothelial dysfunction. And so there's two things right there that your albumin concentrations being low can contribute to your blood clot risk. We started to look at our data, and our serum albumin concentrations, you can see males on the left, females on the right. There was a statistically significant lowering of albumin during flight. It's not that dramatic, and frankly, I'm not going to wave my hands here today to try to convince you that albumin concentrations are lower during flight, because they really don't look all that different. What happened is, when we started to look at the data a little more carefully, one of the things we realized is that combined oral contraceptive use can cause hypoalbuminemia. And some female astronauts will use oral contraceptives to suppress menstruation during spaceflight so that they don't have to deal with that while they're up there. When we look at the female data and break those out by females that didn't use oral contraceptives, which is the line I showed a minute ago, and those that did use oral contraceptives, you can see a significant difference in albumin concentrations. And every one of the 6 crew members in that yellow line had at least one data point during flight where their albumin concentrations were below normal limits. So we maintain what happened here is that combined oral contraceptive use lowered serum albumin. I talked about what happened there before. Inflammatory markers are up during flight, and that can also contribute to lower albumin. Oral contraceptive use will also increase transferrin, which is one of the clotting factor regulators, and that can increase hypercoagulability. I didn't show you the data today, but we do have data that those that use oral contraceptives had higher concentrations of transferrin. We know that spaceflight exposure tends to lead to inflammation. We know that spaceflight exposure tends to lead to change in hemodynamics. So when you put all this together, it shouldn't be a surprise that one of our astronauts clearly had a blood clot, and it's very plausible that others did, that we just haven't seen it. And as a takeaway from this, 2 things. One, I wouldn't suggest that we should not allow oral contraceptive use in astronauts during flight, but as you know, there are different types of oral contraceptives, and specifically the different generation progestins have different effects on albumin. So you can select an oral contraceptive that has a very low or no impact on albumin, while there are others that have a significant impact on albumin, and you need to choose which one you use very carefully. The other point I would make from this, and this is one of the things we highlighted when we published these data, hypoalbuminemia is very well documented to be associated with increased blood clot risk, but this is an often overlooked phenomenon. And most people, as I said, don't pay attention to albumin until it's horribly low, and the concentrations I showed you a few minutes ago were significant, but they were not at the point where you would start to worry about malnutrition or something like that. And that albumin, I think, is an under-recognized contributor to blood clot risk. And blood clots, as we know, affect a tremendous number of people here on Earth. With that, I bring you back to the picture of Columbus's ships in front of the shuttle Endeavour in the background there to make the point that we know that lemons and limes helped to cure scurvy. They didn't cure scurvy through magic. They cured scurvy through nutritional biochemistry. And while many people find it's more interesting to study physiology and not that many people like biochemistry, we always need to remember that it's the underlying biochemistry that causes the physiology to change. And we ignore that biochemistry at our peril. I show you here a couple of the books that we published at the URL there at the bottom. You can download PDFs of those for free. The one came out just a couple of years ago, and that is pretty much everything we know about space nutrition at this point. On the right is the same book, but we built it for middle school kids. It's the same book, it's just got smaller words and better pictures. And again, you can download both of those for free. Speaking of space station, one of the things that is pretty remarkable about space station is that it is big enough that you can see it with the naked eye. If you go to spotthestation.nasa.gov, you can type in your location and it will tell you over the next typically 2 weeks when decent sightings are. And I looked up this morning when the space station will be over Melbourne, and on Tuesday morning at 6:15 a.m. your time, you'll get a 6-minute pass, which is really good. Typically 5 minutes or 6 minutes is as long as you're going to get. It'll appear about 35 degrees above the horizon, and it'll start just above northwest, and it will traverse the sky over the course of 6 minutes and leave over east-southeast. So if you're up in the morning and want to get up and go out, again, it is a pretty remarkable thing. And speaking of pictures from space, I leave you with this, which is a picture of Melbourne taken during the Expedition 47 mission, so probably about 4 years ago, over at Night Pass. Nothing's more incredible than the Earth when seen from space. And with that, again, I thank you for the time and the opportunity to present here, and I'd be happy to try to answer any questions if you've got them. Thanks.
Thank you so much, Scott. That was an absolutely amazing talk. So many fascinating insights. And I can say for sure that there are things that we've heard from you today that we wouldn't hear anywhere else. So it's been incredibly valuable to have you share those insights with us. And thank you also for being so kind as to look up a couple of things relating to Melbourne. And it certainly is really exciting when you do see the International Space Station whizzing by overhead. And that's an incredibly beautiful photograph too, and it was sort of fun to kind of try to pick out what the different landmarks were. So thank you very much indeed. And now we have some time for questions. So who would like to ask a question? I've got a question in the chat there. There seems to be some debate as to the viability of food bioprinting or lab-grown meat in long-duration spaceflight. Some argue that currently these processes lose too many nutrients for them to be efficient, whilst others say it may have psychological benefits for astronauts. What are your thoughts?
Well, I can't speak to that specifically, but I will tell you there are a lot of people out there that have suggestions for how we can do things during spaceflight, and they are typically very challenging. So it's not that we don't think they're good ideas. It's just very challenging from a logistics point of view, from a crew time point of view, to be able to do things like print food or grow food. Or we've had people suggest using cell cultures to grow meat to make a steak. We've had people suggest that we could use crickets to make hamburgers, and all those might be viable. But what you need to remember is it's got to be easy. It's got to be able to fit within the spacecraft. On space station, we can launch food pretty easily. So that's not that big a deal. On the way to Mars, again, it might be great if we could grow steaks from cells, but you need a device to allow you to do that, which means it's going to take space. You're going to need raw materials to do that. You're going to need a way to clean that stuff so that you can do it again and again. And then you need a way to cook it, which we don't currently. We just warm up food. We don't have a way to cook it. So if you could get away with all those things, you come up with a steak, which is one food item. How many days a week can you eat steak? So then you've got to have some sort of vegetable. And okay, then we're going to grow plants and harvest plants. So now you need a growth chamber to do that. You need seeds, you need water, you need nutrients, and all the things that go along with that all become very space constraining, that is volume constraining, and crew time. The amount of time the crew is going to need to spend growing, harvesting, and converting those into edible items is really challenging. And if one of those things breaks, are you going to have a second one in case it breaks, or you're not going to have steak again? And if we're growing everything, we're going to have vegetarians, we're going to fly nothing but plants. That's great. If we lose a crop, do we lose the crew? And those are all the kinds of challenges that we face that make these things very, very hard at least at this point in time. Someday will we get there? We will. Someday will we have a base on the moon where crews will live for months or years on end, and we'll be able to send the material up to built facilities to deal with that? Yeah, we will. Probably not going to be in the next 5 or 10 or 20 years. Is that fair?
Yep, thank you very much. That was a really great answer. And I think you've highlighted some of the key issues that we certainly are going to have to keep developing, keep thinking about, especially as we reach deeper and deeper into space. So hopefully some of these young people can help in coming up with some good solutions for those. So we have another question here. With the rise of personalised medicine, for instance, with the faraway goal of genetic and epigenetic tailored treatment plans, and personal spaceflight measures for each astronaut, how does NASA go about customizing or tailoring their nutritional profiles on an astronaut-to-astronaut basis? Like, what tests do you do to categorize these biochemical and genetic factors? Thank you.
It's a great question. At this point, we're not doing any of that. You could look at our one carbon work with B vitamins as a toe in the door, if you will, in that what we hope is that we will find at-risk individuals that we can give vitamins to help overcome their specific genetic issues. The idea of expanding beyond that, I think there's a little bit done in the space medicine community right now, but really not much, and certainly not on a nutrition perspective. And frankly, I think that's going to have to happen in mainstream medicine before it's easily adapted to spaceflight. At this point, and again, into the foreseeable future, we send up food for everybody, and the crews that go up are expected to eat that food. There have been a few cases of individuals that had specific nutrition issues, like lactose intolerance, for one example, and we've tried to help work around that, applying some extra foods for those crews, because one of the things we found is if you've got a crew member who, for example, is lactose intolerant, if they won't eat any of the foods that have dairy in them, first of all, they're going to eat a lot more of other stuff, and their crewmates are going to be left with all the foods that have dairy in it, and they're not going to have as much access to the other foods. So your dietary issue might impinge upon how well you get along with your crew. So that's one small example, but we, again, do not have the ability at this point to fly foods per astronaut.
Thank you. That actually feeds into something that I was wondering about while you were talking today, and that was how far do dietary issues, like, for instance, you've mentioned lactose intolerance, but also gluten intolerance and low FODMAPs, or having a special dietary preference like being a vegetarian or vegan, does that come into astronaut selection procedures, as well as the potential impact you've mentioned on food availability for the overall crew?
In general, it is not part of astronaut selection, but one of the things that the Foods Lab has maintained for Artemis missions to the moon, and Mars will be the same way, that we will send the food to the moon or to Mars before the crew gets there. So we will not have the ability to tailor it. So while we've had a few instances of that again on space station, it's a little easier to deal with there. But we've said, the foods people in particular have said that the way things are designed at the moment, we cannot support a gluten-sensitive individual on Artemis missions.
Yep, that makes sense. Just two other things that I wanted to ask, seeing as there don't seem to be any other questions at the moment. Do you give crew any routine vitamins, such as vitamin D? So that's the first question. And the other one was with the CHAPEA mission. You mentioned 3D printing out of cement, and I also saw the illustration of it sitting on the surface of Mars, and I was wondering, is that cement design meant to be radio protective against the higher levels of radiation on the surface of Mars? So those were the two things. Thank you.
On the first, we do provide vitamin D, and that is the only thing we provide what I call programmatically. That's the only thing that we provide for every astronaut. If astronauts want to take other things, some will take a multivite. We typically tell them that if they want to, that's fine. If they do take anything more specific, we usually take a look at it to make recommendations of whether or not we have any concerns about it. As you know, some of the vitamins or some of the more specialty vitamins can have very high doses, and you want to make sure that you don't get too much of those. A standard multivite, our typical response to them is that we don't think it's going to help, but we don't think it's going to hurt. The CHAPEA habitat was not specifically built for radio protection, but I know the company that we contracted to build that has sites for putting a device on the moon and someday putting a device on Mars that would be able to take the regolith of the soil from the planet and make buildings out of it. I don't know if anybody's looked at radio protection. Obviously, cement wouldn't be bad, and suppose if you made it thick enough, you might be able to get some radiation protection. But again, I'm not aware if anybody's actually looked into that specifically.
Thank you very much for that. Really appreciate you answering my questions as well. We're almost at the end of our time. So could we all please say thank you very much to Dr. Smith for generously coming along today. We really, really sincerely appreciate it. It was such a great session and really interesting questions as well. So thank you.
Just one sec. Let me show you one more thing. So this is an image from an earlier presentation. This is what that habitat looks like. So on the left, you can see this is them 3D printing it. And so the device over here is spitting out red tinged cement. The gentleman there has a tablet that he's driving the computer, and you can see it's laying down. It always looks like icing to me, the next level of the cement. And then here you see what it looks like. I think, I'm not sure exactly where that is, but this is the door that you go in. The crew bedrooms are here. There's a shower and bathrooms here, and the living areas in here. And then this image is what it looked like when the walls were complete. And then they constructed the ceiling outside and actually moved it in with cranes. But I actually took the two pictures on the left there, and it was pretty mind-boggling to see this thing coming together.
Wow, that's amazing. Thank you so much for showing us that. It's sort of 3D printing on an industrial scale. I certainly wouldn't have thought about that. You're used to the really small ones. It's really fascinating that they can do it on such a large scale and create something so functional.
Indeed.
So thank you again for coming along today. Really, really appreciate it. It was very generous of you and a really wonderful talk. Thank you.
Thanks. Appreciate it again. Thanks for the invitation.
You're very welcome. Thank you.
Take care. Bye-bye.
Bye.